So I'm very pleased to introduce the first speaker of the afternoon session, Professor Martin Brazier, who's a professor of paleobiology at Oxford. Amongst the duties that he's had, he's the chairman of he has been the chairman of the Faculty of Earth Sciences and also the chairman of the Subcommission on Cambrian Stratigraphy. Professor Brazier is perhaps best known for his work on earliest life and the unique lessons that have been learned from the early fossil record, including the Ediacaran biota and the Cambrian explosion, but I would argue also very famously for his work on Foraminifera. And I say that with just a little bit of bias because I also work with Foraminifera, but very pleased to introduce Martin Brazier.
Thank you very much, Mark. It's a great pleasure and mixed with sadness, I suppose, to be here this afternoon and to reflect on some interactions that I've had with Lynn over the last few years, and to begin this session on microcosm to the cosmos. And that is the picture shows the last time I met with Lynn, which is with Betsy Dyer, who's also in the room, and my daughter, who's a scientific translator, and we had a very agreeable meal and a beer, as you can see, in Berlin for a protest conference. Now, Lynn, much to my delight, spent a year in Oxford, 2008 to 2009, when she was invited by Balliol College in Oxford to be Eastman Professor for a year, which meant she had a wonderful house. She dined and lived in grand style in an Oxford college, and she joined me in teaching. So I was able to get to work with her very closely. It was a very happy, agreeable, and I'd like to think fruitful period. Oxford, you may remember, is famous for being the home of Richard Dawkins. So Lynn was highly delighted to actually get onto the home turf of Balliol, of course, was Richard Dawkins' own college. So when Lynn arrived in Oxford, it was like Attila the hen attacking and taking Rome. And she did so with great aplomb, and we had several very enjoyable interactions. And Richard actually was very gracious in the debates we had. The centerpiece for science in Oxford is this wonderful old building called the Oxford University Museum. It's the Natural History Museum, and it's famous, of course, as the place where the first public debate on evolution took place in 1860, with a meeting of the British Association for the Advancement of Science, and the meeting took place on that corridor at the top that you can see there. But I'd really put this in to celebrate something that Lynn drew my attention to in the museum on my own doorstep, and that's the skeleton of that elephant. Now, Lynn got very excited by this elephant because it's an elephant that had died in a circus act in the 1880s and then had been buried in a pit, which became known as the Elephant Pit. And it was in that pit that a previous young female scientist, Lillian Gould, had described an amoeba called Pelomyxa. And Pelomyxa plays quite a large role in some of Lynn's thinking. It's an amoeba without a mitochondrion. And of course, Lynn placed that very low down in the tree of life. So that poor old elephant there has had its own place in the history of trying to understand the organelles. Also, while Lynn was with us in Oxford, she was very busy on some of the books we've been hearing about, the Kozo-Polyansky book, and of course the Kingdoms and Domains book that Michael Chapman talked about. But it was also the occasion when she received a great honor to any scientist, only handed out every 50 years or so, the Darwin-Wallace Medal at the Linnean Science Society on Darwin's birthday in 2009. So I was delighted to be there alongside her when she received that great accolade. But for me, really, the great reward of having Lynn join in with our own group was that she took part in teaching and promoted our thinking. I run a course which Lynn joined in, which looks at revolutions in deep time, symbiotic revolutions, and each of these is strung out along the geological record here. And at the bottom, you can see the geological timescale going from left to right. And in black is shown Precambrian time, which is about 80% of Earth history. And the little golden bit, the golden age at the bottom there, is Phanerozoic time. And Lynn, amongst all biologists, was very, very aware of the enormity of Precambrian time. And it's into that time slice that we're going to fit most of the major evolutionary events within the history of the cell over the last 4,500 million years or so. Each of these major revolutions, I would say, has involved a symbiosis, and certainly the third one, what Lynn would call the protoctista, involved a really important series of symbiogenesis. And I was working with Lynn at the time of her death on a manuscript which had been through many versions. And part of my point of being here now is to say, please, can we get this story moving forward, hopefully with Lynn, also on the paper. And she became fascinated with the story I was telling about Foraminifera, as Mark has referred to there. It's something that had disappeared in my own horizons, but she dug it out and said I really had to start to develop it. This is the last version of the manuscript. She would get usually as far as page 1 or 2, and it would be so covered in comments that she felt it had to come back to me, and I would then be changed, and I'd send it back, and it would still come back in somewhat similar condition. This is a typical intense, careful scrutiny that Lynn paid to abstracts and to the power of words. She was very, very well aware of that. But I'm delighted to say, or rather almost saddened to say, that I've got a book that celebrates Lynn and a whole host of other people involved in the exploration of the eukaryote cell. Comes out, Oxford University Press, in May. It's my little publicity plug. It's called Secret Chambers, and she was a great stimulator of helping me to put that story together. And in fact, that's more or less what I'm going to show you here, exploring the fossil record of symbiosis and symbiogenesis, particularly through forams. And one of the things I've done in this and a previous book is to develop, and Lynn really liked this way of looking at it, that science is not a belief system. It's a unique system for the measurement of doubt. And that means to say that all the things that I might be telling you this afternoon, I'm not saying they're my belief. You're perfectly entitled to challenge them. And what we really need to do is assemble the evidence in an empirical way. I did my doctoral thesis on Foraminifera, presumably like Mark and a number of you here, in Caribbean coral reefs. I spent a year working as a ship's scientist on board HMS Fawn, sampling and dredging the reefs and lagoons around the Caribbean. Large amounts of tropical sand are made from Foraminifera. And they take on the most wonderful sculptural forms. You can see a number of them here, little spiral forms full of little holes. And it was our job to really try to get to understand Foraminifera and their evolution in deep time. There's one particularly wonderful foram, lives in the Atlantic, called Discosporina, which Lynn really enjoyed. And it's this one. It's about 1 centimeter across. And you can see in the center, there is a little round blob, and that's the first formed chamber, and then it spirals out into a sort of spiral coil, and then gradually the chambers change shape from being longer to shorter, and then wider and wider, and eventually there's enormous great fanning out of chambers, and then they're subdivided by partitions with a whole series of multiple apertures. And this foram is quite remarkable because it captures every main stage of foram evolution since the beginning of the Cambrian. There's not many organisms that recapture so much of an evolutionary story. Just to show you that, you have stretched out Discosporina here against geological time, and you can see how its early chambers are what we might expect to find in the Cambrian, in the middle, somewhere in the Carboniferous, and up the top there, the sorts of things we might expect to find in the later Mesozoic and the Cainozoic. Now that's just Discosporina. We can assign different numerical values to these patterns of architecture, because what's actually happening here is that the line of communication within every chamber is getting shorter relative to volume. It's maximizing its capacity to react and conserve energy through its process of growing larger and larger. And in order to get at how this has happened in the fossil record, I devised a thing called the parsimony index, which is shown at the right there, and we can assign a numerical value to the architecture of the mature specimen of different types of foraminifera. Now, this is where it gets interesting, and this is what Lynn was still getting her head round in the manuscript, and this is a story that hasn't yet been published, but where I hope to draw you in. This pattern from long to short lines of communication has happened in many different lineages, and here are at least 40 foraminiferal lineages shown through the last 350 million years or so. And in most cases, they start from long and simple, and these lineages move towards higher values of PI, that is to say, with those short lines of communication and lots of multiple apertures. Very often they do so in the way of forming discs at the left, or spindle shapes in the middle, or sort of Chinaman's hats or cones at the right-hand side. Notice, though, on the diagram at the far right, the fact that the forms that have reached these very complex architectures reach enormous size. It's not unusual for fossil foraminifera to be over 100 millimeters in diameter, and some of those are over 200 millimeters in diameter. These are protists, remember, conceptually single-celled organisms, eukaryotes. But also shown there in red are the points where these lineages become extinct, and I've tallied them along the right-hand side there, the different lineages, and each of those extinctions correlates with a major mass extinction event. What's actually going on here is that foraminifera have repeatedly self-organized themselves from quite simple towards complex forms of architecture, and that very large size has been accommodated by changes in the architecture of the kind we've seen. And the majority of those I've shown you were almost certainly symbiont cultivators. Here's some modern ones that are cultivating diatoms, or dinoflagellates, or red algae, or green algae. And all those that cultivate diatoms and these other algae do so by changing their architecture in order to create cultivation chamberlets. Well, these rules we can extrapolate back to the fossil record. Here's a fossil one from the Eocene, which we're pretty sure was a diatom cultivator. Here's its appearance in the rock. Geologists know about these. This is the group they use to correlate and understand tropical carbonates around the world. But rather delightfully, and Lynn loved this particular paradox, the rocks in which that particular foram, called nummulites, occur are the rocks that make the Sphinx. So you actually have a chimera, which is a term Lynn loved to use about symbiogenesis, two creatures combined, not only in the Sphinx, but the Sphinx itself is built from a chimera which was symbiotic. And I think that is just such a lovely story. And there's one other lovely layer to this too. These little fossils we're looking at in the Sphinx here, nummulites, were the first fossils ever described by Strabo, the philosopher, in 20 BC. He had a very logical interpretation. He thought they were fossilized lentils from the slaves' lunch packs, which had accumulated over the eons. It was a wonderfully logical, cautious interpretation, and full of doubt. Now, if we sum up all the patterns of extinction of these great symbiotic lineages, we can see that symbiosis comes and goes. It builds and collapses many times. And very often before the major extinction events, symbiosis has absolutely reached its apogee with very diverse and very large forms indeed. I think that tells us about how mass extinctions actually work and what's going on. There are also intervals here, NE, no evidence. That's where we have no evidence for major symbiotic associations. And they are interesting periods in Earth history too, like the lower Carboniferous and the Triassic. These patterns of extinction match almost exactly the stories of extinction in reefal ecosystems themselves, which I've drawn here as a roll call, a roll of honor, as though from a battlefield, going from the top in the Ediacaran down to the bottom, in which I listed all the major reefal forms with typical things you might find on a tombstone, such as, "And we shall never forget them, ever in our thoughts. They gave their lives that we might be free." All of it nonsense, of course. But nonetheless, these creatures are no more. Those genomes have disappeared. And it's happened episodically, and I think we really need to get to the grips with that. I did some modeling on this with Boolean networks to show how such collapses might be the function of specialization and how things gradually drive themselves towards more and more specialization through time. But I won't linger on that. Lynn said it was all modeling and therefore rubbish, and she would have none of it, and there would be none of it in the paper, so you didn't see that picture. But she was very interested in an interval I've called the boring billion, which is from 2 billion to 1 billion years ago, here seen in this outcrop in northwest Scotland, area where I've been working on the rocks with Paul Strother, who's also here. Between that period, 1 billion years long or so, extraordinary things happened in the fossil record, and we start to see things through the middle and towards the end of it, which we can confidently ascribe to eukaryotic groups, most of them algal, like these we've just described from lakes 1,000 million years old. So the boring billion, I think, occupies a very interesting phase between the great mass extinctions and the big carbon isotopic wiggles at the top of the diagram there, and an earlier phase of glaciations about 2.5 billion years ago. The boring billion is, Lynn hated this word, by the way. She thought it was an insult to what the eukaryotes were getting up to during this time. It actually refers to the carbon isotopes, and it was a prod or a poke at geochemists that this was a very, very flat and uneventful field in terms of the isotope geochemistry. But to me, it indicates that there was an interval here in the boring billion. There were no glaciations. We know of no major meteorite impacts. I think blue water ecosystems are very widespread. And the sorts of conditions in which symbiogenesis could have built may have been extremely widespread for a surprisingly long period of time. Sort of thing we have not seen in the last 500 million years. Lynn's own diagram, you've seen 2 or 3 times here already, starts to calibrate her great tree of life here against the Proterozoic, and that's the boring billion where most of these things are happening. And what I'm really talking about is mostly the chloroplast, of course, could be the mitochondrion as a major symbiotic event. At the right is a little sketch just to remind you that the chloroplast itself may have undergone multiple endosymbioses, being swallowed up a number of times, first a cyanobacterium, then a red alga or a green alga, and ultimately a brown alga to give something like the dinoflagellates. And all that seems to have happened, as far as I can tell, within the boring billion interval or thereabouts, back in the Proterozoic. So it's a very remarkable interval, because what we're seeing here, in effect, is during that period in the Precambrian, symbiosis got to the point where wholly new types of cell were constructed. But what I've been showing you in the Foraminifera is that symbioses build, but then they very typically collapse, and that there's no real convincing evidence, as far as I'm aware, that new cells have been built during the last 570 million years or so. It's as though there are 2 different ways for evolution to have been working in deep time. And I paraphrase it here a little bit, that symbiogenesis, by which I would mean the development of organelles, may be a distinctively Proterozoic phenomenon, and that the transfer of information and the building of new organelles may have been brought about by very distinctive conditions, as shown by the green bar. When we move towards the Cambrian, and certainly through the Phanerozoic, we're seeing the build-up and collapse of ecosystems and symbioses, and mass extinctions are things we see here, which we do not see in the Precambrian. And we have fashioned the term symbioclasis, the breakdown of major symbiotic associations, as a distinctive feature of the later Phanerozoic. But science, sorry about the bar there, science is a unique system for the measurement of doubt. So this is just a hypothesis. It's not a belief system. And with that, I'll finish my talk. Thank you very much indeed.